my great honor and privilege to introduce Dr. Susana Carmona, who is an inspiration to me as a scientist and also increasingly becoming a dear, dear friend. She directs the Neuromaternal Research Group at Gregorio Maranon Health Research Institute in Madrid, Spain. Before that, she earned her PhD in neuroscience from the Universidad Autónoma in Barcelona. With the support of numerous international fellowships, she spent 6 months as a PhD student at Columbia University and completed postdoctoral training at Harvard. In addition to running her own research group, Dr. Carmona holds a number of senior leadership positions, which is really just a testament to the standing in her field. She leads the neuroimaging platform at CyberSam, which is a biomedical research center headquartered in Madrid. She co-chairs the Pregnancy and Parenthood Working Group at the Enigma Neuroendocrinology group with Magdalena Martinez. And she is a lead scientific advisor for us, for the Anna S. Bowers Women's Brain Health Initiative. So Dr. Carmona's primary research focus is characterizing the brain changes that occur during pregnancy and into the postpartum period, into motherhood. She is just truly a pioneer who has led numerous national and international projects in this field. She's authored over 50 original scientific publications, including features in Nature Neuroscience, Nature Reviews Neuroscience. She just had a beautiful cover just last month. And she recently published a book for the general public summarizing all of her main findings in the field. I cannot wait for it to come out in English. I'm so eager to read it. And I do want to just say on a personal note, she is an incredible artist. I've had the pleasure of seeing some of her work. The cover of Nature Neuroscience is actually an original production of Susana, and from her trainee Magda, who's now a postdoc with us, I've gotten to see even behind the scenes a little bit more of her work, and it's truly just stunning. I want to just mention a few things too about that are coming up to put it on everybody's radar. Dr. Carmona is going to host the upcoming Parental Brain Conference in Barcelona. This is in May of 2025. This is in partnership with Oscar Villaruela. So everybody should sort of Google that and put that on your map. And I'm super excited to attend this year for the first time. And she is going to be an inaugural visiting faculty fellow for the WBHI. She's joining UCSB in August. So everybody that's in Southern California, will get a chance to meet with her. So without further ado, it is my great pleasure to welcome Susana Carmona for her lecture.
Thank you very much, Emily. Let's see.
While she gets her slides up, I just wanted to point out if people have questions, they can enter them in the Q&A box. And I think we'll wait until the end just for flow purposes, because we'll be releasing this on YouTube later. So if anybody has a question, just enter it there and we'll get to it at the end. Thanks.
Okay, well, first, thanks a lot for giving me the opportunity to present here in this wonderful seminar series about women's brain health. It is a real pleasure. I hope I can do justice to this introduction. And today's talk, I'm going to focus on the neuroanatomical changes associated to humans, but I will also provide some background about what we know in non-human animal models, because this research has really paved the way for us to advance in humans. And I'm going to take the liberty of starting by the take-home message, the conclusion, because by the end of the talk, I hope I have provided enough evidence to demonstrate you that the transition to motherhood produces profound, dynamic, and long-lasting adaptations in the human brain. And also that we have preliminary evidence suggesting that these adaptations might facilitate maternal behavior and mother-infant bonding, and that they seem to be modulated by gestational hormones. Okay, let's start. So, as you know, pregnancy is a very common phenomenon. Around 100 million women around the world give birth every year. But despite being very common, it is also an extraordinary phenomenon, physiologically speaking, because I would say it's the most extreme physiological event a human being goes through, because in just 9 months, almost every organ of the woman's body adapts to facilitate gestation, parturition, and postpartum. There are changes at the cardiovascular level, the respiratory level, the gastrointestinal level, the musculoskeletal level. So for instance, blood volume increases by nearly 50%, oxygen consumption and tidal volume also increase. The immune system suffers super interesting changes that dynamically adapt to tolerate this genetically different organism in our body. Well, those of you who are mothers know that even the color of our skin changes in certain parts. So there is like a whole change in nearly most of the organs of the woman's body. But pregnancy is not just about adapting the organs that we already have. Pregnancy also entails the creation of a new organ, the placenta. So the placenta acts like an interface between the mother and the fetus, exchanging nutrients and oxygens, but it's also an endocrine organ that synthesizes and secretes hormones that travel through the bloodstream and go to the different tissues, indicating the organs of the mother that they should now function under a different homeostatic status, under a different equilibrium or range of parameters, so everything works fine for gestation, parturition, and postpartum. Well, there are a lot of manuals describing each of these adaptations and how to treat them in case something doesn't go as expected. But surprisingly, we're just starting to discover what happens in the brain during pregnancy and which potential implications these changes might have on the neurological and mental health of the woman at the short and at the long term. And this is even more surprising if we take into account that there is a lot of evidence indicating that hormones, especially sex steroid hormones, are powerful neuromodulators. These hormones can bind to specific receptors in the different brain cells and induce a series of neuroplastic mechanisms. But let's review what is neuroplasticity. So neuroplasticity can be defined as the intrinsic ability of the brain to adapt in response to internal or external stimuli. And this can be translated at the cellular level in different scales. So for instance, we can find changes at the molecular level that affect the synaptic transmission between the neurons, to changes at the morphological level, or even the number of brain cells, not only neurons, but also glial cells. Of course, we all maintain a certain degree of neuroplasticity during our life, but there are periods in which the brain seems to be especially plastic, especially malleable or switchable to learn. And one of the most paradigmatic examples is adolescence. So we know that the abrupt increases in sex steroid hormones during adolescence coordinate a series of cellular changes such as myelination and synaptic pruning that are so huge that can be also detectable, traceable at the macroscopic level with MRI. So with MRI, what we found during adolescence is that gray matter volume decreases, and decreases in a consistent manner with cognitive development. So the regions that are in charge of more basic functions, such as the motor cortex, are the ones that show this decrease earlier. Presumably because these changes, these gray matter reductions, reflect a fine-tuning of these circuits, the maturation of these circuits. Well, this is what happens in adolescence, but what about pregnancy? Sorry. So this is the hormonal storm during pregnancy. I would say that pregnancy is like the quintessential hormonal storm in the human being. So there is no other period in the life of a human being in which his body and his brain is invaded by all these hormones that we know are powerful modulators. We have, for instance, increases in prolactin, estradiol, cortisol, progesterone during gestation, also increases in oxytocin around parturition. And some of these increases are around 1,000-fold. For instance, I think estrogen is like 1,000-fold. And then after parturition, these hormone levels drop drastically with placental expulsion. So this is more or less what we can see at the hormonal level. But as I have mentioned, we have barely no information about how this hormonal storm might affect the brain of a woman during this period. We know nothing or nearly nothing about humans, but fortunately, we have more than 50 years of research in non-human animal models. And from this literature, there is consistent evidence that gestational hormones induce certain neuroplastic mechanisms that facilitate maternal care. So I will briefly review this literature because, as I have mentioned, this literature has paved the way for us to advance in humans. And I will start by defining maternal behavior. So maternal behavior in rodents, it's a change in the behavioral repertoire of the animal dedicated to ensuring the physical well-being of the offspring. And this change in the behavioral repertoire really fascinates me in rodents because the animal passes from ignoring, avoiding, or even attacking the pups to suddenly becoming super interested on the pups and wanted to interact with them. They find them really rewarding and want to be in touch with them and learn from their cues. And these changes in the behavioral repertoire can be detected before the birth of the pups, such as, for instance, nest preparation, but the changes become really evident once the pups are present and the animals start showing all these behaviors such as nest retrieval, breastfeeding, licking, grooming, and also protective behaviors such as aggressiveness towards the strangers. And all in all, these behaviors increase the chances of pup survival. I'm going to show you some videos extracted from a publication by Bianca Marlin at Columbia University, just to display this switching behavior from being non-maternal to being maternal. This is the behavior of a virgin female mouse in which the experimenter places a pup inside the cage. And you will see that the animal basically ignores the pup. And this behavior is very different from what you will see here. That's the behavior of a mouse that is a mother. So as you can see, the animal here, the stimuli of the pup, finds the pup, and retrieves it to the nest. But what is even more surprising is that these researchers found that if they inject oxytocin in certain parts of the brain of this virgin mouse, they induce a certain neuroplastic mechanisms that can facilitate the learning of maternal behavior. So you will see here, in this case, you will see a naive virgin mouse in which the experimenter has injected oxytocin in the left auditory cortex and after cohousing 12 hours. So this virgin mouse now retrieves the pups and brings it to the nest. So presumably what oxytocin does is to increase the saliency of pup stimuli and thus enhance or accelerate the learning of retrieval behavior. Okay, so let's see what we know about the maternal circuit in rodents. So we know that gestational hormones, especially late gestational hormones, link to specific receptors in the rat's brain, and in particular in the region known as the medial preoptic area of the hypothalamus. And from this, two main things happen in relation to pup stimuli. On the one hand, there is an inhibition of the avoidance or aggression circuit, and in parallel, there is an activation of the reward or motivational circuit. And once the first is inhibited and the second activated, the pups become really motivating and salient stimuli for the mother, who will search to be in contact with them, will interact with them, and will learn from them. And from that, pups will further shape the circuit and adapt maternal behavior to their own needs. And importantly, this maternal circuit can also be activated in virgin males and in virgin females and males through this continuous exposure, through the interaction with the pups. So it's a little bit slower. I think it takes around a week. And recent evidence in mice have managed to refine so much that they can talk about specific neurons, no longer about the medial preoptic area of the hypothalamus, but a specific set of neurons known as galanin neurons that are basically neurons that express the neuropeptide galanin. So in a set of experiments, the group of Catherine Dulac from Harvard University used fiber photometry recording and optogenetic activation to demonstrate that these neurons are essential for parental care by reducing parental aggression and increasing parental motivation. I don't have time to show you the videos, but I really recommend you to look at them. They are linked to this study. because it's amazing to see how researchers can turn on and off maternal behavior just by manipulating these neurons. And they can do so not only in females, but also in males. Yeah, so this author, Johannes Koll, recently from his own laboratory now at Kirk Institute in United Kingdom, published together with her postdoc, Rashida Marie, this publication on October last year. So in this publication, what they found is that estradiol and progesterone during pregnancy program these galanin neurons so that they respond more persistently, strongly, and selectively to PAP stimuli. And they do so in a very specific manner. So estradiol seems to silence the noise of some of these neurons and enhance the activity of those that will respond to PAP stimuli, while progesterone will rewire the circuit in a permanent manner. Another super cool article, in this case about the role of prolactin in this maternal circuit in mouse. So actually, nearly 20 years ago, this first publication found that prolactin seems to promote the creation of new cells in the subventricular zone of the animal that then around parturition migrate to the olfactory bulb. And now, in a publication in November last year, researchers have demonstrated that these neurons that become interneurons in the olfactory bulb respond especially to PAP stimuli. So they help the mother to recognize their offspring smell. So to sum up, from non-human animal models, especially murine models, and particularly in rats, we know that hormones and the interaction with the PAPs induce a certain neuroplastic mechanisms in the animal's brain to facilitate maternal behavior. And these neuroplastic mechanisms include differences in protein expression, differences in synaptic transmission, differences or changes in the number of dendritic spines and dendritic branches, also myelinization, and also changes in the number of cells. And as we have seen, some of these changes are long-lasting, while others disappear during weaning. So for instance, those that I told you about, progesterone and estradiol modulating the galanin circuit, seems to be long-lasting, whereas those of prolactin seem to disappear after the weaning of the pups. If you want to know more about the specific neuroplastic mechanisms for the different brain regions and during the whole transition to motherhood, and also about the role of these hormones on these neuroplastic mechanisms, I recommend you reading this review that we published last year, in which especially the first two authors, Camila Servín and Magdalena Teresa, did a great job in summarizing and synthesizing the many years of work of giants in the field of non-human maternal brains, such as Lisa Galea, Jody Pawlowski, Benedetta Leuner, and many others. Okay, so this is what we know from non-human animal models, but what about humans? Are gestational hormones also inducing neuroplastic mechanisms in humans that facilitate maternal care? We'll see. So most of the literature in humans comes from functional MRI studies that focus on the postpartum. So what they do is to scan mothers during the postpartum period, and they use this classical paradigm in which they show mothers pictures of their own baby and compare it to pictures of others' babies. And after many years of research, many reviews, and many meta-analyses, there are a set of neurons that seem to be activated when the mother saw pictures of their own baby. And these include the ones that you can see here, and that we can group into different circuits. So we, for instance, found activation of the mesolimbid dopaminergic circuit and the hypothalamus, which we have seen are keynotes of the rodent maternal circuit as well, and that presumably signal the baby stimuli as highly rewarding, highly motivating for the mother. Then regions such as the insula and the anterior cingulate cortex that belong to the salience network, also known as ventral attentional network. And this network is involved in reallocating our attentional resources towards what our body has signaled as highly relevant, in this case, baby stimuli. and also changes in regions of the default mode network. That is a precuneus, medial prefrontal, and temporal parietal junction. And this default mode network, as you know, is involved in internal processing and self-perception, mind wandering, theory of mind and empathy, all these internal processes. This is a super interesting field of study, but it's not today's, it's not the focus of today's talk, because today's talk, I wanna answer this question. That is, does the brain at this time point, is the brain at this time point different from the brain at this time point? Or in other words, does the brain of nulliparous women change during their first pregnancy? And before I answer this question, I would like to explain you the personal story that is behind this question on why we had this question. And this story is located in Barcelona, Spain, around 2008. By then, I was working as a postdoc at Part de Recerca Biomèdica, and I worked with my colleagues Celine Huxema and Erika Barba. And one day, while I was driving them in my car, Erika told us that she wants to have a baby. We were in our 30s, and she told us that she wants to have a baby. And while still on my car, we started discussing about what we thought we knew will happen to a woman when she gets pregnant. And we didn't fully agree. So as soon as we arrived to the laboratory, we started searching on PubMed to know what will happen to Erika's brain once she becomes a mother. And honestly, we were expecting to find a lot of literature, but to our surprise, we only found the literature in rodents that I told you, like all this literature that backs from 50 years. And in humans, we just found two little pieces of evidence. One that comes from a paper of Angela Otrich in 2002 that compared the size of the brain, how the size of the brain changes in 9 women without preeclampsia and 5 women with preeclampsia. And then also an interesting evidence that found that pregnant women have bigger pituitary gland. And this finding was observed in postmortem studies in 1898 and then confirmed later on, nearly a century later, with MRI. That's all that we found. And we started the same day, we started building on this evidence and fantasizing about the ideal design to answer the question that we have. And we got so excited about the design that at the end of the day, we decided to talk with the PI of the laboratory, Oscar Villarroya, and ask him to perform this study at extra hours and without any funding. And he surprisingly agreed. So we started collecting the data in November 2008. And just to show you the amount of effort it took us to perform this first study, we started data collection on November 2008, and the paper got published on February 2017, so nearly 10 years later. Okay, so what did we do in this study? We contacted participants, we contacted women before their first conception, and then again at 2-3 months postpartum. So we contacted 25 women before they got pregnant, and then again at 2-3 months postpartum. And as controls, we included a group of nulliparous women, scanned at similar time intervals. And to additionally control for other potential sources of variability, such as childbearing, we also scanned their respective couples. So that is first-time fathers and control men. And I'm going to show you the results of comparing these 2 time points, the brain at those 2 time points. And what we found were pronounced gray matter volume reductions only on the mother's group. And these reductions were not randomly located in the brain. They overlap pretty clearly with what we know as the default mode network here in magenta or dark green. This network that I told you is involved in internal processing, mentalizing, and theory of mind, and that activates when the mother saw pictures of their own baby. So once we saw that, we wondered if these changes might be something related to the early postpartum or if they will be long-lasting. And to answer this question, we contacted a subsample of the mothers and scanned them again at 2 years postpartum. And those are the results of the comparison. So each of these graphs represent each of these regions of interest, and in blue you have the period of pregnancy. So you can see that gray matter volume reduces during pregnancy, and these reductions are still present 2 years postpartum for nearly most of the regions, except with for the hippocampus that seems to revert a little bit. This is a very schematic representation of what I just told you. So we found that from preconception to 2-3 months postpartum, we found this decreasing gray matter volume that seems to be still present at 2 years postpartum. But what does that mean? It is related to this personally cognitive differences in mother, this mommy brain or mommynesia. I mean, it's related to cognitive performance. So in our study, we also evaluated a neuropsychological battery that included verbal memory and working memory. And we did not find any difference between the pre and the post session, nor we found any association between the brain changes and cognitive performance in these metrics. But we found evidence that these brain changes seem to facilitate maternal attachment. So in the post session, we asked participants to fill questionnaires about maternal attachment, and we found that the gray matter volume changes significantly predicted the quality of attachment and the absence of hostility. And this, together with all the evidence in non-human animal models in rodents, suggested to us that probably or possibly these changes might facilitate the maternal transition. Okay. So with this study, we answered this question: does pregnancy modify the human brain? Yes, we have shown that pregnancy produces gray matter volume reductions that persist at least 2 years postpartum, and that seem to facilitate maternal behavior. But most importantly, this question opened up a series of questions that we, as well as other researchers in the field, have been trying to answer for the last now 7 years. So for instance, we wanted to know if the brain changes are indeed similar to those that take place during adolescence, if subcortical regions that are key for growing maternal circuit also change, anatomically change during this period, if brain changes reverse or remain after the first 2 years postpartum, what happens to the father's brain, which is the temporal course of the findings, is it linear or is it dynamic? And also if these changes are, as in animals, mediated by hormonal fluctuations. I'm going to start with the first one. Oh, sorry, yeah. So I'm just going to dedicate the rest of the talk to answer some of these questions, but for those of you who want to delve into the details, here I paste the main publications linked to each of these questions so you can take a look at them later. The first question: are brain changes similar to those that take place during adolescence? Okay, so the thing is that the results of the study I told you about had a great media coverage, and sometimes journalists made a statement such as the ones you can see here, like woman's brain atrophy with pregnancy. And when we had the chance to respond, we usually tell them that in biology, as in many other things in life, a decrease in volume or less can be more. I mean, a reduction does not necessarily mean an atrophy or a neurodegeneration. And we use the example of what happens during adolescence, that as I told you, there are decreases in gray matter volume that are consistent with cognitive development. That's what we respond to them, but actually we haven't empirically tested this. We did so 2 years later, in this publication in 2019, in which we scan girls before and after puberty and compare the changes during puberty to those that take place during pregnancy. And we also add a control group of nulliparous women. And in this case, instead of just focusing on gray matter volume, we use surface-based analysis to provide a very detailed description of the anatomical or the cortical changes. No? We extracted whole area, cortical volume, surface area, cortical thickness, and different indicators of gyrification. And here I'm going to show you the results. So on the y-axis, you can see the estimated monthly rate percentage change. And on the x-axis, you have all these metrics that I told you. So total brain volume, cortical thickness, surface area, and so on and so forth. In red, the changes on the adolescents, in blue, the changes in the mothers, and in green, the changes in the female control. So as you can see, the profile of changes in the mothers is nearly identical to that observed in the adolescents. Both pregnancy and adolescence entail a flattening of the cortex with reductions in cortical thickness and surface area. Does that mean that these two processes reflect the same cellular mechanisms? We don't know, because we have to remember we are analyzing the brain at the voxel level, and within each voxel we have around 100,000 different types of cells. So different cellular changes can lead to the same change at the voxel level. But at least macroscopically, we can say that the changes that take place during adolescence are similar to those that take place during pregnancy. So to the question, are brain changes similar to those that take place during adolescence? The answer will be yes. Pregnancy and adolescence entail similar neuroanatomical adaptations with a flattening of the cortex and decreases in cortical thickness and surface area. Next question. Are subcortical regions of the rodent maternal circuit also affected? So far, I told you mostly about the cortical differences, but we wanted to know if subcortical structures that are key for governing rodent maternal care do also anatomically change during the transition to motherhood. So we focus first on the nucleus accumbens, that is a key node of the mesolimbic dopaminergic circuit, and within the maternal circuit, it seems to signal the pups as rewarding and facilitating the interaction with the pups. And to answer this question, we manually segmented the ventral striatum, which is a part of the striatum that contains this nuclei accumbens. And we found that the right ventral striatum is reduced during pregnancy, and the reductions remain at least 2 years postpartum. And we also detected that the more the gray matter decreases in this nuclei, the higher the activation of this nuclei when the mother saw pictures of her own baby. And very recently, a study led by a laboratory of Elseline Huxema, whose first author is Clara Spalek, did a similar procedure, but now segmenting different subnuclei of the hypothalamus, and they found that certain subnuclei of the hypothalamus are reduced during pregnancy, and reduces seems to be associated with estradiol levels and metrics of fetal maternal attachment. Okay, so are subcortical regions in the rodent maternal circuit affected? The answer will be yes. The right ventral striatum and certain subnuclei of the hypothalamus seem to decrease in volume, but in the case of the hypothalamus, this decrease seems to be associated with different indicators of maternal care. Next question, do brain changes reverse? I'm going to recover this schematic representation, now incorporating the subcortical regions in there. So we found these gray matter volume decreases from preconception to 2-3 months postpartum that are still present at 2 years postpartum. But on the other hand, there are studies in large-scale databases, such as the UK Biobank, that analyze the brains of thousands of women, most of them at midlife, that is between 40 and 80 years old. And in some of these studies, they compare the brain of those who are mothers to those who are not. And in a seminal study by Anne-Marie Delange, they found that mothers show like what they call a younger looking brain. So the estimate of brain age in the mother's brain is younger than that in non-mothers. And this suggests that pregnancy leaves a lifelong impact in the human brain. And this lifelong impact has been also observed by other researchers, such as Edwina Ockart, and with other metrics, such as restlessness, fMRI, diffusion, or even with cognition. But we cannot forget that all this evidence comes from cross-sectional data. And to properly answer this question, we need longitudinal studies. And this is what we try to do. We try to contact the participants of the original study, the publication of Huxema et al. 2017, and scan them again at 6 years postpartum. And the first problem that we had was that the sample was very small, because most of the mothers already had a second child at 6 years postpartum, And most of the controls, including myself and Elsadine Huxema, who participated as controls in the original study, already became mothers during this period. So at the end, we end up with only 7 mothers and 5 nulliparous controls. But although it's very small, I want to highlight that currently it's the only longitudinal database that tracks the changes from preconception to 6 years postpartum. And here I show you the results. Here I represent as a, just to remember that the changes we detected from preconception to postpartum to 2-3 months postpartum. And on the right side of the image, I show you the changes from preconception to 6 years postpartum. And as you can see, reductions in regions, especially those of the default networks, such as the medial prefrontal cortex and the precuneus, remain reduced in volume at 6 years postpartum. Here you can see more or less the same now for each region of interest separately. And in gray, you have nulliparous women, and in black, you have mothers. And again, the blue represents the period of pregnancy. So as you can see, there are reductions during pregnancy that are still present at 6 years postpartum. Okay, so to the question, do brain changes reverse? The answer for now will be no. At least at 6 years postpartum, some of the volumetric gray matter reductions in regions of the default mode network seem to be still detectable. Next question, do father's brain undergo similar changes? A couple of minutes ago, I showed you this slide, and I told you that we didn't detect any significant differences in the father's group. But the truth is that the changes in the mother's group were so huge that we use a very restrictive threshold at expenses of missing some positive findings. But when we plot the raw data, we found these kinds of distributions. So those are the percentage, the bootstrap distributions of the percentage of change from preconception to 2-3 months postpartum for cortical volume, cortical thickness, and area. Mothers are depicted in pink, controls are in blue and are centered on the 0. And as you can see, the father's distribution is laid in between the two. So fathers change, but they don't change as much as mothers. And this is suggesting that besides gestational factors, there are also environmental factors that seem to play a significant role on the neuroanatomical changes of the brain. We, as well as other groups, published some data about the father's brain, but I don't have time to go through all of them. But I will refer you to all the publications of Darby Saxby, because she's really the expert on the father's brain, and she has found super interesting associations between the brain changes and testosterone and oxytocin levels, as well as parental engagement. So do father's brain change? Yes, but the changes are smaller than those detected in mothers and seem to be associated with parental engagement. Now, which is the temporal course of the changes? So far, I've told you about studies that compare the brain from preconception to early postpartum, and they found these gray matter volume decreases that seem to be still present at 2 years and 6 years postpartum. But there are another set of studies, longitudinal studies, that compare the brains of mothers at different time points during the postpartum period. And these studies show increases in gray matter volume. So how do we fit all this data together? And by pulling all this data, by pulling all this evidence, different trajectories emerge. For instance, this trajectory in which it is possible that there are gray matter volume decreases during pregnancy that valley at parturition and then slightly recover during the early postpartum, but do not reach pre-pregnancy levels. But this trajectory is also possible. So it is possible that the gray matter reductions that we attributed to pregnancy might indeed be produced only by birth. So in order to better delineate this gray matter volume trajectory, we perform a study focusing on the peripartum period. That it's where we are missing most of the data. And this study got published on January this year. And what we did was to scan 110 pregnant women at the third trimester, and then again during the first month postpartum, and control women at similar time intervals. And to be completely sure that our findings are robust and reliable, we included a replication sample, an independent replication sample of 29 pregnant women and 24 control. So I'm just gonna mention the results that are replicated with this independent sample. So the first thing we did was to compare the brain cross-sectionally. So we compared the brain of pregnant women and nulliparous at this first time point. And we found decreases in gray matter cortical thickness and volume. Here I just displayed the cortical thickness, but very similar changes are also detected in cortical volume. So we found decreases in cortical thickness, indicating or suggesting that these reductions are present before the birth of the baby. And to our surprise, these cortical reductions were not confined to the default mode network, but they were widespread around the cortex. Next, we performed this longitudinal comparison. We compared the brain longitudinally, and we found that some of these decreases seem to attenuate during the early postpartum, and that the higher the percentage of postpartum time between the sessions, the higher the increases in cortical thickness, which is suggesting that an inflection point around birth that seems to slightly recover during the early postpartum. An additional analysis indicated that this recovery rate is different depending on the functional network. So decreases that, I see like some comments on the chat, I hope it's not for me. So decreases that belong to the default and frontoparietal network seem to recover at a slower rate. And all in all, this is suggesting a trajectory compatible with this one, with gray matter volume decreases that seem to ballet around birth and slightly recover during the early postpartum, but do not reach pre-pregnancy levels, at least for certain brain networks, such as the frontoparietal network or the default mode network. And probably this is why we also found default mode network decreases at 2 months postpartum, 2 years postpartum, and 6 years postpartum. And while this suggested U-shaped trajectory has been now, it's no longer a suggestion, it has been also observed in a recent publication by the group of Emily Jacobs using a dense sampling study. So in this study, Elizabeth Krustel then sampled her brain and hormonal levels from preconception to 2 years postpartum. I think she underwent a total of 26 MRI scans. So this is a very comprehensive study, and they provide, they delineate the trajectory of white matter integrity and the trajectory of different subregions of the hypothalamus. But here, I'm just gonna focus on the trajectory of cortical thickness, because as you can see, they observed this beautiful curve of gray matter volume decreases that seem to ballet around parturition, slightly increase during the early postpartum, but do not reach pre-pregnancy levels. So to the question, which is a temporal course of the findings, we can say that now we know that pregnancy entails decreases in gray matter volume that seem to ballet around parturition and slightly recover during the early postpartum, but do not reach pre-pregnancy level, as I have mentioned. Next question, are these brain changes mediated by hormonal fluctuations? Well, we have we have solid evidence from rodent studies indicating that gestational hormones induce neuroplastic mechanisms that facilitate maternal care. But the truth is that in humans, data is still preliminary. Oops. I'm sorry. I hope I can finish the talk. Okay, so I'm going to review the the emergent literature we have. So besides the data I told you about the relation between the hypothalamus, subnuclei of the hypothalamus and estradiol levels, the group of Elceline and Huxema in 2022 published this study in which they compare the brain from preconception to early postpartum. And they found that gray matter volume changes from preconception to postpartum were associated with third trimester levels of estradiol. However, this study does not include intrapregnancy data, intrapregnancy MRI data, so it cannot fully capture the dynamism of these variables, hormonal fluctuations and brain changes. On the other hand, we have the study, the then sampling study that I just mentioned, that found that the gray matter cortical gray matter volume changes are concomitant. They coincide temporally with levels of estradiol and progesterone, but again, this is just N1, so it only scans one subject. And to properly answer this question, we might need larger sample size that also include intrapregnancy measures. And this is what we have been working on together with a group of Oscar Villarroya in a paper that is now under review, in which we scan 127 pregnant women and 52 controls, including among these controls gestational and non-gestational mothers. And we scan the brain from preconception to 6 months postpartum, including intrapregnancy metrics. And we found that the trajectory of gray matter volume changes and the trajectory of estriol 3-sulfate and estrone are significantly associated. So to the question, are these brain changes mediated by hormonal fluctuations? I would say that changes seem to be mediated by estradiol levels, but we still need more evidence. Of course, as we advance in the field, more questions arise. Some of them, you can see some of them here, and we are already trying to, to answer some of these questions. For instance, we might need to fully characterize the brain at the multimodal level, including diffusion weighted images, rest and state, spectroscopy, and so on and so forth. It will be interesting also to track the relation between these brain changes and not only endocrine factors, but also immune factors and immunoendocrine interactions. Also, we might need to test the relation between these brain changes and microbiota changes or changes at the vascular level. We need to know if some of these changes also happen in non-gestational mothers, what happens in subsequent pregnancies, if the brain also changes in second pregnancies, for instance. If some of these changes can help us to predict and treat postpartum depression, as well as to know the relation between the brain changes and the genetic profile of the participants, race, ethnicity, gender, socioeconomic status, or even parental leave policies of the country they live in. But if you want to know about how the field is heading now, I recommend you reading this publication we published like a month ago, and whose first author is Magdalena Martinez, that was a PhD student at my laboratory, and it's now a postdoc at Emily Jacobs' laboratory. And in this comment, Magda proposes the use of two complementary approach to advancing the field of the human maternal brain. On the one side, using then sampling studies that give priority to the number of time points over the number of subjects to gain statistical power, and also population-based studies that do the opposite. They give priority to the number of subjects over the number of time points. And these two approaches will help us to optimally answer some of these questions. So for instance, then sampling studies are great at capturing within subject variability, therefore will be ideal for answering the questions at the top, for tracking the dynamism of these brain changes and how these relate to other factors that are also highly dynamic within the same subject. And on the other hand, population studies are great at capturing between subject variability and therefore will allow us to better approach the questions at the bottom, such as if these brain changes are related to the genetic profile, race, ethnicity, gender, socioeconomic status, or parental leave of the parental leave policies. And yeah, so the idea is also to call for a collaboration, because it will be fantastic to start creating this large-scale database probably arriving to a population level of dense sample individuals that we can track around the years or across the years, and we can leave this database as a legacy for future researchers in the field that want to answer questions such as, for instance, if reproductive history impacts on the menopause, or if reproductive histories have something to do with the risk of certain neurodegenerative disorders. And I would like to wrap up by recovering the take-home message, because I hope that by the end of the talk, because I hope that now I have already provided enough evidence to demonstrate that the transition to motherhood produces profound, dynamic, and long-lasting adaptations in human brain anatomy, and also to let you know that we have preliminary evidence suggesting that these neuroanatomical adaptations seem to facilitate maternal behavior and mother-infant bonding, and seems to be modulated, at least in part, by gestational hormones, especially estrogens. So I want to conclude by thanking all the authors whose work I have discussed in this presentation, and also the Women's Brain Health Initiative for allowing me to contribute to this wonderful series. And here I let you my email address and also the name of the social media channels of our group, because those have been super useful, not only for the dissemination of our studies, but also for the recruitment of the participants. So I encourage other researchers to use these channels to facilitate disseminating their findings, but also contacting potential participants to participate in the study. And that's all from my part.
Susana, I'm always blown away by your talks because they're so comprehensive and are just, again, a testament to your strength in the field. I have so many questions, but before I jump in, I want to give the floor to the Q&A. So Rebecca asks, I work in the field of psychiatric genetics, so I'm curious if you have any brain scan data for postpartum anxiety or depression.
I don't have, I know there are publications that have scanned psychiatric patients during the postpartum depression. I haven't included this data here, but it's a tremendously interesting field of research, especially now that we have this new drug, this thuranolone, that is a metabolite of allopregnanolone. And this is also highlighting the necessity of studying how these hormones influence the brain during this period. But yeah, I know there are many publications about that topic. It's just that I haven't included them here.
And then one question I have is, right now, I think a lot of the data that we have available to us is based on kind of a high resolution anatomical scan and a resting state scan. Are there, and I ask because this audience tends to, what we're hoping to cultivate is really a mix of kind of the leading women's health scholars and also those that are kind of pushing methods development, both in terms of data acquisition protocols and kind of from the analytic end. So if you could wave your magic wand and say, I wish I had this sort of data, or I could work with this sort of group who is an expert in data analytics, what would you want to have, or what sort of collaborations would you want to spur, and how can we make it happen?
What a difficult question, and what an interesting question, Emily. With you, that's what we are doing, right? So we are trying to collaborate and with you and many other experts in the field of neuroimaging. So one of the questions we have now is to disentangle the cellular mechanisms behind these brain changes, and also it is important to take into account that we are studying the brain as an isolated thing, but it's inside a body, and the whole body changes during pregnancy. So any technology that allows us to integrate all this information, physiological information, will be it's necessary.
Mm-hmm.
I have a quick question, actually. This is very specific. So the UK Biobank study that did this classification, and they found that younger looking brains predicted if a person had a child or not. And I wondered about the apparent mismatch, or maybe I'm missing it, between the two things. So is it perhaps that even after 6 years, you have a flip in the thickness Because I would assume that if a younger age prediction would mean that you had thicker gray matter, but maybe
Yeah.
I'm missing the
No,
point.
I mean, that's what I thought at the beginning too. It's like what the logic might tell me. So the thing is that these algorithms are like kind of black boxes. You don't know really what they are predicting. But yeah, you're right. So reducing cortical thickness, like intuitively, you might think it might predict an older age, but there are different possibilities. And one of them, and I'm now talking about a review about cognition of Edwina Ocker, they found that at the long term, this cognitive demand of childbearing kind of switches. So you have been like training your brain for a lot of years, and then suddenly the demands of your child disappear and your brain has been trained. So when you get to the older age, there is like a flip. But honestly, we still don't know which are the specific markers of this younger looking brain. I think they are now working on identifying these markers. And also the lifestyle at the long term, the potential lifestyle of childbearing might, I don't know, lead you to a lifestyle.
It's kind of related to my other question, which was about stress and sleep, because having a child, like carrying a pregnancy to term and having a child involves, you know, as you said, all these hormonal changes, but there's also a huge lifestyle change that occurs, and you can see it in the fathers, right? So the fathers who haven't carried the child to term, but they do have these brain changes. So it'd be really interesting to see those non-gestational mother studies, because of, you know, stress and sleep and huge lifestyle changes can cause these maybe as well.
Yeah. So Celine, on the last paper that I told you, she discarded the effect of sleep and osmotic effect as well. But yeah, it's interesting. And before I forget, also there is a very interesting hypothesis that with regard to why they might look younger, and it's about, so there is data suggesting that reproductive history kind of adapts the inflammatory state in which the person enters to the menopause, and this might be like a protective effect for dementia. So, but yeah, there are many factors, and of course, I always want to mention that we are looking at the very small part of the brain, but the brain is inside a body. But we are in a society, so there are a lot of factors that might shape this parental brain.
And then what's happening for baby number 2 or 3 or 4 while you're going through that while caring for a whole brood? Oh my goodness. I only have one, so I can't speak from personal experience, but I can only imagine.
Yeah, me too.
We have 1 more minute left. I don't know if there's any other questions, or if Jaren, you wanted to make an announcement about the next.
Yeah, definitely. We have also like 2 more questions we want to ask, or I can share my screen next songs.
Yeah, so thank you for pointing that out. Carolyn asks, are there studies about the evolution of other cognitive abilities other than verbal and working memory, so other executive functions, both short and long term?
Yeah, so the field of cognitive, the cognitive changes during pregnancy, it's also super interesting. And again, I'm going to refer to this review by Edwina Ocker, because at the end, what they show is that there are certain, so meta-analysis, we haven't found any association or any difference during pregnancy, but meta-analysis really indicate that there are decreases in memory during the 3rd trimester and during the 1st year postpartum. But other studies seem to talk about that this range, like these reductions of memory, are specific to tests that do not involve the baby. So when they ask for items to remember items about the baby, mothers used to perform better. So I think that there is still a lot to figure out about these potential changes, but around 85% of women refer they have memory losses during the 3rd trimester and the 1st month postpartum. And I think there we have like biological factors, psychological factors such as switch on priority, and also societal factors of what confirmation bias that we have about what might happen to us.
And one last question, because I want to squeeze it in. It's such a good one from Ina Stelzer. Amazing talk, Susana. Do you see any influence of pregnancy pathology, preterm birth, preeclampsia on brain dynamics?
On our data, we haven't we haven't included any patient with preeclampsia, but it's interesting. But the first publication I told you, the one that we found when we did this PubMed in 2002, Angela Otrich, actually they came across similar findings by accident because they were really comparing preeclamptic women with women without preeclampsia. So at least at the macroscopic level, they seem to show the same trajectory.
Thank you so much for joining us. I just want to remind everybody to follow Susana's group on social media channels. She is such a huge public voice in this space. She's amassed a huge following, as you can say, for good reason. So thank you again. And July 8th is our next seminar series. We're going to be moving into the world of sort of neurodegenerative diseases with Dr. Caitlin Castelletto, who's joining us from UCSF. So we'll see everybody next month. Thank you again, Susana. Brilliant talk.
Thanks, bye.
Thank you. Bye.
It was a great talk. Thank you so much. Bye-bye.
Bye.
Exceptional talk. Thank you. It was wonderful. Bye.
Bye.